This is GATE 2022 Life Sciences Section P Biochemistry. Question number 40. Purification of 6-his stack protein using nickel NTA column is an example of dash. Okay, you must all be familiar with nickel NTA column, nickel nitriloacetic acetic acid column. Okay, and these are the different chromatographic techniques. This is the NPTEL online course reference for the different chromatographic and biophysical techniques in biochemistry. So we had already studied this before. I'll briefly go over these chromatographic techniques here once again. So first we have a ion exchange chromatography. Right, in ion exchange chromatography, the column is packed with either negatively charged species or either positively charged species, depending upon protein of our interest. Okay, if we want to separate positively charged proteins, specifically we'll use negatively charged we'll package with negatively charged beads, Cipharos or agarose beads, to separate positively charged proteins, and vice versa. To separate negatively charged proteins, we'll use positively charged Cipharos beads. Okay, and after the mixture of protein solution is passed through these column. Okay, the protein of our interest here, protein of our interest is a positively charged protein. It will get bind to these negatively charged exchangers here, and when we want to elute, we'll add a washing after adding this, we'll add a washing solution which will wash off the excess binding which are false positively bound to this, and then finally we will start eluting the protein of our interest using a specific salt concentration of elution buffer. Okay, so we'll elute out our positively charged proteins. Here it is getting eluted in tube number 3 and 4. Similarly, in size exclusion chromatography, we have different sizes of Cipharos and agarose beads packed, and these polymer beads are porous in nature. By porous I mean they are the different proteins, the different sizes of protein can pass through, right? So the smaller size of protein will pass through this porous polymer bead, and it will take a longer time to elute, whereas the larger beads these larger beads will slide from the surrounding of these porous polymer beads, and they'll elute out faster. Okay, so most likely the larger size proteins will elute in 1 to 3 tubes, right? The bigger protein here as shown, they are eluting in the 3-4 tubes, whereas the smaller proteins will elute out very late, and we don't need to add any elution buffer here. We just have to monitor different protein, and we can monitor this using different techniques. Okay, then let's come to affinity chromatography. We are discussing this for the first time. So in affinity chromatography also, we have a chromatographic column which is packed with a polymer bound. It is packed with NTA polymer bound to a polymer. So these are the polymer beads I am talking about. Our column is packed with these beads. Okay, and to these beads we have a special molecule attached, which is nickel NTA. This is nitriloacetic acid. Okay, which forms a complex with nickel 2+. Okay, and this is very important. This type of affinity chromatography is very important technique used in molecular biology and biochemistry to purify his stacked protein. Okay, what are the his stacked protein? So in lab we express different types of protein, and these proteins we engineered them to be histidine tagged. So generally the protein are coded in such a way that the N terminal or the C terminal of the protein has 6 histine residues. Mostly they are 6, but it can also vary to 11, and the minimum histidine residues can go down below 3 or 2. Okay, mostly 3. In very rare occasion we have 2 histidine or so. Okay, so this histidine tag could go till 11 histidines, and this histidine forms a complex with this nickel. Okay, nickel NTA. So this complex is very strong complex. The affinity is also very strong. So suppose we want to purify a recombinant protein. We have a recombinant protein which has a his tag in a mixture of a protein isolated from a E. coli or any organism. So in this mixture of protein we have a histidine tagged, and in the affinity chromatographic column we have this polymer beads which has nickel NTA. Okay, so this nickel NTA is packed in the column, and when we add the mixture of proteins, okay, what will happen? The protein of our interest which has the histidine tag, it will get bound to this nickel NTA beads. Okay, and then we add a washing solution. Washing solution will wash off the excess unbound or positively or false positively bound proteins to this nickel NTA beads. Okay, and after the washing step we'll start eluting. Okay, the unwanted proteins obviously will get washed through the column as shown here, and then we'll add any solution or any elution solution. This elution solution will have higher affinity to the nickel NTA beads than that of our protein, and due to this higher affinity it will elute our protein and it will itself get bound to the nickel NTA beads. Okay, so from 6-7 tube we'll get a protein of interest. This is very strong chromatography technique used to where it is very frequently used to isolate the protein of our interest. So generally whenever we are isolating protein from an organism, we can use series of chromatographic techniques. Like we can first start diluting our protein samples by doing size exclusion chromatography to obtain the protein of our interest, and then we can do either ion exchange chromatography or affinity chromatography, depend upon which protein we have. Okay, and finally this is also very important type of chromatography technique known as hydrophobic interaction chromatography. It is also known as HIC. So what happens in hydrophobic interaction chromatography? Suppose our protein of interest has a hydrophobic patch. Okay, and this hydrophobic patch in a protein, in any protein, the hydrophobic residues are stuffed inside the protein, right? So the protein is for the protein mostly to be soluble, all the charged species will be surrounded on the outside of the protein, and the hydrophobic residues will be inside. Okay, this is the hydrophobic effect, right? All the hydrophobic residues will be facing each other in the core of a protein, whereas the hydrophilic residues will be outside the protein, and they'll interact with water, and thus the protein will have a higher solubility. Okay, so here also we have a hydrophobic interaction column, right? This is the hydrophobic interaction column, and this column has a ligand molecule attached to this on the side of this column. So this ligand molecule, so we are interested in hydrophobic patches of the protein, right? So this column is tightly layered by the hydrophobic ligands, which are mostly alkyl ligands. Okay, so these ligands are butyl, octyl, heptyl ligands. So these alkyl chains are hydrophobic in nature, right? CH2, CH3 molecules, these all alkyl chains are hydrophobic in nature, and these are bound to the side of the hydrophobic interaction column. Okay, now in a solution, suppose we take a solution of suppose in a beaker we take our protein of interest. Okay, this is our protein of interest, and to this beaker we'll add any salt like ammonium sulfate. Okay, NH4, like ammonium sulfate, NH4, and ammonium sulfate solution. So and then what will happen when we start increasing the concentration of ammonium sulfate in the protein solution? What will happen? The ammonium sulfate will pull out all the water molecules, and the outside of the protein will be devoid of all the water molecule, and this will force the protein to be completely open up, and pull out all the hydrophobic patches from the inner core to the outside of the protein, right? And this process is known as salting out, right? When we add salt into a protein, what will happen? This salt is obviously charged. It is both positively and negatively charged, and it will take up all the water molecules with it. As we increase the salt concentration, it will take up all the water molecules along with them, and it will also take the hydration shell of the protein. What is hydration shell? Hydration shell is the water surrounded by the water which is surrounded to a protein. So as I mentioned earlier, the protein is protein charged residues will be outside, and these charged residues will attract water. So there will be formation of a hydration shell around the protein, and this hydration shell is completely taken by the salt when we start increasing the salt concentration in the solution. So this hydration shell will be completely removed as we go on increasing the salt concentration. Okay, and in absence of this hydration shell, what will happen? The hydrophobic residues which are situated in the core of the protein, it will come out. Okay, they'll force them to come out. Now what will happen? The protein of our interest will have a hydrophobic patch on the surface of the protein, right? It will have a hydrophobic patch on the surface. So this is black, which is protein of interest. It has a strong hydrophobic patch. So when we add the protein solution here, it will first bind to the hydrophobic patch, and we'll have some residual protein binding also here, okay, which we do not want. Okay, so what we'll do, we'll add a washing solution. And what is washing solution? This washing solution will be lower molarity of salt concentration. Suppose here we are using a salt concentration of 10 molar NH4Cl. Here washing solution will have a salt concentration of 5 molar or 2 molar. Okay, this washing solution will remove the excess which are not that tightly bound protein from the column. They'll remove all these proteins, and finally our protein of interest will be tightly bound to this irrespective of the washes we give. And the final step is the elution. Okay, this is the general chromatographic steps. Final step is elution. For the elution step, we'll again use very low molar concentration of salt, right? We'll use very low molar concentration of salt, And this process where we are decreasing the concentration of salt, this process is known as salting in, right? This is very important. Salting out, we add excess of salt, right? We add excess of salt to remove the water, and in salting in, what we are doing, we are decreasing the salt concentration in the solution so that there is reappearing of the hydration shell around the protein. And now since the hydration shell has reappeared, what will happen? The hydrophobic patches will again go inside the core of the protein. So denatured protein will again become renatured, right? The protein will form its own native structure again, and since there is no hydrophobic patch on the surface, it will get eluted. So this is how hydrophobic interaction chromatography is used. This is very important technique. So these are the few advantages why we use hydrophobic interaction chromatography. So first is large amount of sample can be loaded, right? This hydrophobic interaction chromatography is generally used as a starting step of protein purification. So during the starting step of protein purification, we have large volume of sample, okay, and that large volume of sample, it will be very difficult to be loaded in ion exchange or size exclusion chromatography. It will take us very longer time to get the protein of our interest. So first technique we use to do protein purification is this HIC, right? Hydrophobic interaction chromatography. Also samples with high ionic strength can be used, right? Like I mentioned, this technique is well suited before ion exchange or gel filtration and affinity chromatography, right? Purification steps generate very large sample volumes that can be coupled with method. Same the first point. It helps in purifying large volume of the sample, and it can be clubbed with ammonium sulfate fractionation. So it can be clubbed with salting in and salting out using ammonium sulfate fractionation technique, right? We all know what is salting in and salting out using ammonium sulfate. And this sample can be used in ion exchange chromatography step, okay, right? The next step, we can do either ion exchange or affinity chromatography. So the purification of 6-his stack protein using nickel NTA column is the correct answer is A, affinity chromatography.